occurring in multiple places on that particle. And we have transfer, we have multiple redox steps within one enzyme. So these are features of catalysis that so far as we've encountered, are unique to this enzyme. Soon in the TCA cycle, we will encounter another enzyme. Works pretty much just like this. And there are other examples in the cell as well that are organized as these big particles. Yeah.
Is this pyruvate dehydrogenase complex catalyzing a ton of different reactions? Like there's 60.
Yeah, that's right. Yeah, it's happening all over the surface of this. So there might be some regulation around one E1, E2, E3 unit talking to another one, so that maybe they aren't going with the same efficiencies. I'm not that familiar with how, to what extent. It might not be all 60. It might be half of them. It might be, but for sure they can go on. Many of them go on at the same time. Yeah.
So to stop this from happening, we need to make the disulfide bond. We need to stop that nucleophilic substitution.
To stop it from happening?
Right. To stop this reaction from happening, we want to stop that nucleophilic substitution by not reducing FAD.
Yeah, I mean, if I wanted to stop this from happening, I might just withhold NAD. That would be, we go one round, that would be it. Right? For example. This is also a regulated enzyme, as we'll see. So while the reaction is occurring on multiple subunits, multiple places on the particle, it's also subject to allosteric regulation by different molecules. So we'll see that as well. Any other questions about PDH or PDC?
In the diagram, I'm seeing a lot of, I'm assuming the names which are trying to point to the structures rather than a part of the structure that's just being named rather than drawn out. And like the
Hydroxyethyl TPP?
Yeah, sorry, I have a weird angle here.
Yeah, sorry.
So is that a different structure that's attached to there, or is that the name of that structure?
That's the name of that structure.
Okay, cool.
Yeah. All of these are names. Yeah. When they're pointed like that. Here, the name and the name is symbolizing the structure. The structure is not drawn. Other questions? Okay, so we go on from here, and you see here that the top is acetyl-CoA, so there's the product of our PDH reaction. And it enters this cycle, right? So this, of course, is quite famous in the annals of biochemistry. It's called the tricarboxylic acid cycle, or the citric acid cycle, also called the Krebs cycle, abbreviated as the TCA cycle. Hans Krebs is the biochemist who first discovered it and recognized that biochemical pathways could be cyclical. It wasn't appreciated before, that could actually happen. But what we have here is 8 enzymes in a cyclic pathway by which glucose is ultimately going to be fully oxidized, and fatty acids are also going to feed into this and become fully oxidized. Fatty acids are going to be broken down, as we'll see later, into acetyl-CoA. So once we've learned about this one time, we've learned about it as far as the breakdown of pretty much anything in the cell that's going to yield ATP energy. Okay, so sometimes cells even break down amino acids to yield ATP energy. Usually that occurs if the cell is quite hungry and is more of a starvation state, because proteins, if they're broken down and used for energy production, proteins have structural roles in the cell, so it usually means the cell is eating itself up. But there are conditions in metabolism where proteins can get broken down and be used for energy. But mostly it's sugars and fats. Fats get broken down to acetyl-CoA, sugars get broken down to acetyl-CoA. So we've got 8 steps, and you can see that the CO2 steps, this is quite complicated, so you have to stare at this for a while. So there's no way out of that. But Here's CO2 coming off here, and here's CO2 coming off here. So the CO2s are gone in steps 3 and 4. Acetyl-CoA comes in, and it's important to pay attention to the fact that these carbons that come in are labeled pink. So these carbons came from pyruvate, and before that they came from glucose. So you can imagine that they might be labeled in some way. So it's possible to radioactively label substrates at particular positions or at all positions. So for carbon, the usual isotope that's used is C14. So we could say, well, carbon is labeled C14, or glucose is labeled C14 at a particular carbon, right? So then what happens to that carbon as you go through a pathway, right? So that's a way of looking at where things go, right? So these carbons, of course, are coming from glucose, and if you go back through glycolysis and look at it, you could pinpoint where in glucose these carbons are coming from. And so here they are again, right? The first reaction is a condensation between acetyl-CoA and oxaloacetate to form citrate, which is what the cycle is named after. This is now 6 carbons. Okay? 6 carbons, it's rearranged in this reaction to form still 6 carbons, citrate to isocitrate. It's just a rearrangement. Here's the OH up here, and here's the OH down here. So it just got moved around. Now, and then you have your 2 oxidative decarboxylation steps. And note that after these decarboxylation steps, that label, that acetyl-CoA here, let's imagine that you have C14 in those carbons, it would still be C14 here. Right? The CO2s that left were from this molecule, from a previous turn of the cycle. So what comes in as acetyl-CoA doesn't get oxidized right away. It has to go through the cycle a couple times, and then it'll get oxidized. So here it's pink, and here there's no pink anymore. Right? Everybody see that? So what this means is that the C14 here has been what we call racemized into the other carbons. That means mixed up. And so what that means is that we've formed a molecule here that is absolutely symmetrical. Right? So when it goes on to react in the next step, 2 of these carbons were labeled, but when it binds to this enzyme, it's going to bind one way or the other, and there's going to be nothing to distinguish which way it binds because it's an absolutely symmetrical molecule. Right? So that means that the label, by the time it gets here, the label is effectively distributed among all 4 molecules. Right? And so when you get to here, among all 4 carbons, when you get to here, you could say, well, these 2 are still these 2, and I know which these are and these are, right, for one particular molecule coming off the enzyme. But if you look at the reaction in bulk, and you have many of these succinyl-CoAs, you would know where the carbons are. But once you get to here, and you have 5 billion succinate molecules, and you ask for any one of them, where's the carbons, right? They're going to be either in these 2 or these 2. And then it's going to go on. It's going to be the same issue. So you can't tell anymore. Everybody clear about that? How that works? When you lose the label, you lose the capacity to identify which carbon is labeled. Right? So you could say that they're all labeled at this point. Does that make sense? Okay. That's what I mean here. This is a little bit awkwardly expressed. Can no longer denote where the acetate is. I mean, you can no longer denote where the C14 carbons that were originally part of acetyl-CoA. You don't know which ones they are anymore. What else about this cycle that's of general interest? You can see that there's 3 steps here that are generating NADH. One of these enzymes, by the way, has an isozyme that can use NADPH, but we won't worry about that for our purposes. And then there's one that generates FADH2, and then there's actual generation of GTP and therefore ATP, effectively, in one step. So that's a substrate level phosphorylation. So that's just like in glycolysis, we have 2 of those, right? And here we've got one again. So that's these 3 NADHs and 1 FADH2 per acetyl-CoA coming in, right? So Therefore, you can multiply everything by 2 if you're coming from